I hope it's not a problem. I started the YouTube stream and sometimes there's feedback because we have multiple streams going on my computer now. I didn't even think about it, but it shouldn't be a problem.
Yeah, I don't hear anything.
Yeah, so far you sound good. Yeah, sometimes it's weird loops that happen. It's kind of
I think if I just keep the Zoom meeting in the front.
Okay. Yeah, so far you sound
Otherwise, if not, then I'm just going to mute and Megan, you can do the introduction.
Okay. Great idea to do the live stream. That's good.
All right. Well, we'll get started. And thanks to all of the Zoom people for coming. And it's my pleasure today to introduce Erik Sotka, who is a professor at the College of Charleston in Charleston, South Carolina. So this is one of, again, the benefits of having a Zoom seminar is we can invite people pretty much from anywhere, as long as they're on the right time zones, I guess. And he has worked in marine systems, received his PhD in 2001 from the University of North Carolina in Chapel Hill. And previous to that, received a Bachelor's of Science in Zoology from the University of Washington. He's been at the College of Charleston since 2005. And before that was a postdoc at Hopkins Marine Station at Stanford. And I think without going through all of his awards and all the stuff that he's done, we're just going to jump right into the seminar. And so welcome, Erik, and we look forward to the seminar. Thanks for coming.
Thank you, Paul. It's nice to be here. I appreciate the invitation. I was going to talk about some work that we've been doing in our lab with a Spartina salt marsh plant, and which is native to the southeastern United States, and an invader, a red seaweed that comes from Asia, and sort of use these as case studies of what I see as a really fascinating field, which is ecoevolutionary feedbacks, ecoevolutionary dynamics. And so today, good thing, bad thing, rapid evolution of a native salt marsh and an introduced seaweed. So coming up in about 2 weeks is Charles Darwin's birthday. Charles Darwin I wanted to start with, both because of the impending birthday, but also because he is instrumental in everything we do in biology, and certainly forms the basis of much of my thinking for today's talk. I also, on a personal basis, really relate to Charles Darwin. Whenever I see a statue, I try to either, I show him my love. And there's a few reasons why that. You know, Charles Darwin was first and foremost a naturalist. He, like many people of his time, really had, the only tools they had were observation, using their eyes, their sense, and knowing about the world. And that basis really formed much of his thinking for the rest of his career. And I also worked on a species that he worked an enormous amount of time on, which is barnacles. Marine barnacles have this fantastic life history in which they're benthic as adults, and then they pelagically dispersed as juveniles. And he thought a great deal about variation within and between species in these animals that I really loved and spent most of my postdoc thinking about. I guess the other reason I have an affinity for Charles Darwin is that he's very human. And, you know, when he released or allowed the release of his journals, you know, you'd see all kinds of scribbles that I could envision us in pandemic 2020 maybe writing as well. So things like on October 1st, 1861, but I am very poorly today and very stupid and hate everybody and everything. I think, you know, we've all been there, we all understand it's a very human emotion, and it makes Charles Darwin, I think, seem more relevant and real to us. Another one, and you know, I'm not proud that I have these feelings as well sometimes. I'm going to write a little book for Murray on orchids, and today I hate them worse than everything. Anyway, that was not a good day for him. And yet he would persevere through actually a great deal of strife in his life. And he was obviously formed the foundation of the theory of evolution by natural selection and descent. And, you know, this is actually what we think of as the first evolutionary tree that he wrote in his journal in 1837. Today, with the explosion of technology and DNA sequencing, we've been able to expand this tree, whereas you can, and to the point where you can download these evolutionary relationships from places like the Open Tree of Life. Thousands of species are placed onto this tree to give us a sense of origin and ancestry. So much of his thinking about natural selection, I would argue, is about the role that ecology played in driving selection. So if we think about, say, the Geospiza system, these finches are either seed eaters or they're insect eaters, and those that divergence based on the ecology led to speciation events. And so one way to think about Charles Darwin then is that the ecology in these species, in these populations, drives evolutionary change. Now, what we've recognized is that there is also the possibility of the reverse, that rapid evolutionary change can actually influence the ecology of the environment, the abiotic and the biotic environment. And so there is a huge push now in the field to understand, when does evolution itself influence the ecology? We're all familiar with examples of very rapid evolution. You can think of tuberculosis that's become resistant to our strongest antibiotics. I have to deal with lice that are on my children every once in a while, and they're resistant to over-counter medicines. We know that there are herbicide-resistant plants and so on, and this can happen at very small timescales. In fact, I teach my evolutionary biology students that it only takes a single generation to be able to detect evolution. And so there's a movement now to think about not ecological timescales and evolutionary timescales as short versus long, but really thinking of it as when does evolution potentially influence ecological processes? And so that's one big question. And then the other question that I think is even more important is what are the consequences of these? Is there changes in ecological processes that yield differences in ecosystem function, ecosystem services, and so on, things that we might actually put a value judgment on as being good or bad in terms of conservation? Okay, so I'm going to talk today about rapid evolution in two estuarine species that occur here in South Carolina. One is a salt marsh, and the species that dominates these salt marshes here, Spartina alterniflora. And then the other one is a species of seaweed called Agarophyton vermiculiphyllum. Okay, to start with then, I want you to place yourself into the southeastern coastline of the United States. We do not have any rocky shores. Instead, we are dominated on our shorelines by barrier islands that shift with the sands. And inside of our estuaries, we have the interface between the ocean and the land in these salt marshes. Now, these salt marshes are dominated, particularly in high salinity areas, by a single species called Spartina alterniflora. This is a remarkable species that's able to tolerate full-strength seawater because it actually has salt glands that are able to remove the salt from the water and allow them to be able to live. And they're pretty unique in this environment. It actually gets outcompeted in more fresh environments by other wetland species, but in high salinity areas, it becomes basically a monoculture of this one species because of its salt tolerance. It's an incredibly important ecosystem. If you look at data on the amount of acreage of this one plant, it's over 1 million acres between North Carolina, South Carolina, and Georgia. And within every one of these salt marshes, it has an incredibly important foundation species role. So it's highly productive. It generates much of the carbon and nitrogen cycling that we see in these estuaries. It prevents erosion of the shoreline. It buffers eutrophication. And importantly, it's also a nursery habitat for a lot of the species that we like to eat, the shellfish and the fishes. Now, ever since the first description of Spartina alterniflora in the 1800s, people have recognized that there are two basic morphs. Down next to the low marsh, that is at the lower elevations next to creeks and the estuarine water bodies, we have what we call the tall form Spartina. It's, as you'll see, taller than what we see in the mid or upper marsh, which is what we call short form Spartina. Now, the differences depend between short form and tall form depend on exactly the environmental context, but in general, when I walk out onto a marsh, I see tall form that goes between 1 meter to 2 meters tall, whereas in the short form, it tends to be less than 1/3 of a meter, about 1 foot to 2 feet. So huge differences in height that have been noticed for at least 100 years. Now, most of the thinking about this cline is that moreover, these tall form and short form plants cline into each other as you move away from the water. This is a student at the College of Charleston named Daniela Ajunta. She and I did a transect from next to the water and then went about 15 meters away from the water. And one thing you can see is that we have a pretty steady decline as you move away from the water. So tall form and short form may actually be a clinal variation, but for today, we're going to think about the extremes in our experiments and our DNA analysis. Moreover, there is a shift in density as you move away from the water. So next to the water, you have very sparse densities, but if you do have a plant, they're very tall. Whereas away from the water, you have very high densities, but those plants are short. And I'm going to come back to what seems to be a trade-off between height and density in a bit. Okay, so the questions that I'm going to answer today are the following. What is the role, if any, of rapid evolution in maintaining this tall versus short form Spartina? Most of the literature basically assumes, and some experimental evidence support the notion, that the differences have to do with phenotypic changes or phenotypic plasticity from the environment. What we're trying to do is actually show that there's also a genetic basis to the plasticity, as well as a genetic basis that can explain the differences between the tall and short form. Secondly, is there phenotypic evolution? We're going to use a common garden experiment and a reciprocal field transplant experiment to try to detect that. And then, is there genetic differentiation at some molecular markers that we call single nucleotide polymorphisms? Okay, so the common garden experiment. In 2014, we collected plants from 3 marshes in South Carolina. Here we are in the southeast of the United States. Here's my hometown here. And then we have inside the harbor at one site, another site where there's a creek bed, and then right behind a barrier island. In each of those zones, we took plants, seeds from plants in the tall and the short zone. They were separated by somewhere between 20 and 100 meters. And we reared those in a greenhouse to gain a sense of, if you just kept them in a single, very pristine environment, benign environment, then what is their stem height? And indeed, when they were kept in this greenhouse at a constant benign environment, the seedlings that were reared from seeds collected in the field were taller than those from short zone plants, those seedlings from short zone plants. Now, I want you to note the y-axis here. The difference is not that profound, and especially relative to what we see in the field. So the difference was statistically different. We had 30 to 38 centimeters difference after over a year in the greenhouse. But in the field, we had plants that were nearly 1 meter in the tall zone. So one thing to note is that this experiment by itself is not going to completely explain the height difference between tall and short form in the field, but it does suggest that there is a genetic component to that plant height. Okay, so the gold standard for being able to tease apart environmental and genetic influences on phenotypes is to do a reciprocal transplant experiment. So we took seeds that we collected in the field, and some of those seedlings we used for common garden, but we also maintained some of the clonal replicates, and we reared them tall enough so that we could out-transplant them into the field and follow their course over a growing season. So this was a massive effort. My collaborator, Randall Hughes and Tori Hanley at the Northeastern University, and in particular, Robin Zarabecki, who's a PhD student at Northeastern University, really were the drivers of this manipulation. And they dragged all these plants from Boston all the way down to South Carolina in a U-Haul. Outplanted them, and we followed them for about 6 months. And when we look at plant height, we see a very strong interaction between transplant zone and the origin of those plants. So just to orient you here, on the x-axis, we have the zone in which we put the plants, right? So the short and the tall zone. And then the black line represents the reaction norm for tall zone origin plants, and the red line represents the reaction norm for the short zone origin. There was very little difference in plant height in the short zone between those two genotypes. However, there was a profound difference between the genotypes at the tall zone. And in particular, the tall zone genotypes grew taller in the tall zone, whereas the short zone genotypes were unable to grow taller. And that's going to become important as we start thinking about implications for the functioning of these ecosystems, especially in the face of sea level rise. Now, this was all above ground height. We also took the below ground root mass and compared the biomass in the below versus the above ground biomass. And the genotypes from the tall zone were about equal between when we place them in the short and the tall zone. But notice that those short origin genotypes, because they were unable to grow tall, their above ground ratio was very low. And so in fact, you had more below ground ratio. So more below ground biomass. When we look at stem density, there was a very strong environmental component to stem density. So in particular, the tall zone and the short zone plants both had much more stems in the short zone, whereas they both had very low density in the tall zone after a growing season. And that's consistent with what we saw in the field. Remember, the short zone were more dense, the tall zone were more sparse. In terms of total biomass, then, there was no difference between the genotypes at the short zone, but the tall zone, because they were able to grow taller than the short zone plants, they had more biomass in total. So this yielded a significant interaction between transplant and origin, but really what drives stem density and total biomass is not necessarily this interaction, but a strong environmental component. Okay, so summary thus far then. There's a significant genetic by environment interaction in plant height, allocation to above versus below ground, and then stem density and total biomass were actually less significant, but still we saw a significant pattern in total biomass, suggesting that for those that have a genetic component, those traits that have a genetic component, there is the possibility that evolutionary change maintain those differences. Now, the implication for us is that we know that sea level rise is going to threaten these Spartina salt marshes, and much of the literature has been focused on the upper end. That is, in order for Spartina salt marshes to be able to persist in a rising sea, we have to be able to allow them to migrate inland. And there's a lot of problems there, because when you migrate inland, you're migrating into backyards of houses, or you're migrating into a forest. One of the things our data suggests is that we also need to think about the fact that at the lower edge, the salt marsh needs to keep from drowning by evolving consistently high ability to grow tall, right? So these tall form ecotypes need to keep up with rising sea level at the front edge of the Spartina marshes. And that rapid evolution has not been, to our knowledge, incorporated into many of the models of how sea level rise is going to influence salt marshes moving forward. Will these salt marshes be able to evolve quick enough to keep from drowning at the front edge. Okay, well, the question that always kept popping up in my head when I was thinking about the evolution of this system is, well, are these tall and short form plants representative of very local evolution that happens repeatedly, or do they represent historically separated lineages that have invaded these marshes and then sort themselves out across these tidal heights? So another way of saying that is, what's the evolutionary history of these ecotypes? Is it that there's cryptic evolutionary lineages that were separated historically, and then they now co-occur on a single marsh? Or alternatively, are they lineages that, that evolved independently across marshes and in parallel? So you got the evolutionary divergence happening at a marsh, and that evolutionary divergence happened between marshes. In an independent manner. And so the way to answer that question is to look at the population genetics of these systems to get a sense of that demographic history. Our approach was to genotype single nucleotide polymorphisms. So we collected plants from 11 marshes from Florida to New Hampshire. So that's going from the Gulf of Mexico all the way into the Gulf of Maine. At 6 of those marshes, we collected tall form ecotypes that were right next to creek or open water. So we asked people, or we did ourselves, collections less than 2 meters from the creeks, and we called that the tall form. And we also had some measurements that indicated that they were indeed tall form. Then we also asked people to collect plants from 20 to 200 meters away, and that were in short form areas. We collected 15 to 20 plants for every one of these ecotypes and locations. Each plant was 1 meter apart. We used a RAD-seq approach, a restriction associated digest approach, which you have basically subset the genome. It's a reduced representation of the genome, and then we identified polymorphic loci using Illumina sequencing. If anyone's interested, I can talk to you about exactly what we did. But what we ended up with was across 309 individuals that sequenced well, we had over 2,700 SNPs that we are analyzing, largely using genotype likelihoods. Again, I can go into those details for anyone that is interested. Okay, so the first result I want to show you is to convince you that these loci actually worked, that the genotyping worked. And to do that, we looked at broad biogeographic analyses of these polymorphisms. So you can see the map has the marsh locations that we collected all the way from Florida to New Hampshire. And when we do a PCA of the genotype likelihoods using PCA and GSD, we see a split between these populations that's reflective of very ancient divergence, and that has in the literature been recorded for chloroplast DNA as well as microsatellites. So there's a group between South Carolina, North Carolina, and Florida that's on the left side of axis 1, and then a group in New England, right, Rhode Island, New Hampshire, and Massachusetts, and on the right side of PC1. And then PC2 separates the South Carolina, North Carolina group from Florida. So again, these are broad biogeographic patterns that we see in the literature. But the question of real interest is what happens within each of these marshes. And what we see consistently is that there is divergence in these genetic loci between short form and tall form ecotypes. This is one example from South Carolina. Short form is in red and tall form is in black. So again, this is an analysis just of what's happening in a single site. But this pattern repeated itself across 5 of the 6 marshes that we looked at. So on the left, we have PCA for, say, one marsh in Sweeney, Massachusetts. On the right is an admixture plot, which uses linkage disequilibrium and to cluster our genotypes into 2 groups. And those 2 groups very cleanly split between the tall, short divergence. So at Sweeney, West Marsh, Coal State, Bowen's Island, and Folly, we see divergence between tall and short form. And again, these plants were collected between 200 and 200 meters apart. St. Joe's Bay is a place where we did not see divergence nearly as strong. And one of the things that Randall Hughes tells me about this bay is that you don't see profound short versus tall form at St. Joe's Bay. Next to the water versus inside the marsh, there's not nearly the morphological divergence. All right, so what does that mean then? What that suggests to us is that there's been repeated evolution independently at these marshes between tall and short form. Now, the question then arises, well, why? Right? And we don't know, and this is work that we're trying to think of moving forward, and would love some input from anyone. The first is that in every generation, there's going to be strong purifying selection, as particularly in the tall zone for tall zone ecotypes. And the reason we think that is that it turns out that survivorship is very low in the tall zone. So this is a plot from that reciprocal field transplant of survivorship on the left and the proportion of flowering stems on the right for, again, short and tall transplants, and then again, short origin and tall origin seedlings. You can see survivorship was about 30% for both tall and short form. In the tall zone, survivorship was about 90% in the tall zone. I'm sorry, in the short zone. Similarly, the tall zone actually did flower more than did the short zone ecotype in the tall zone, but it wasn't by much. So we think if there is selection happening at a local scale, it's likely extremely strong for plants that are able to tolerate inundation by the seawater, as well as being able to get out of the seawater at high tide. And that's driving the divergence between short form and tall form. That's one hypothesis. The other hypothesis is that there's a difference in phenology between short form and tall form. So if we have differences in flowering time, then what that's going to do is maintain geographic differences between that might evolve for other reasons, like for local purifying selection. Now, we don't have a lot of data for this. The literature is suggestive that some marshes flower earlier next to the creek relative to in the middle of the bed. This fantastic student, Daniela, went out to 3 sites for an entire growing season, flowering season actually, and every week marked the frequency of flowering. And what she found is that, similar to the literature, some sites the short zone was earlier than the tall zone. Some sites the tall zone was earlier than the short zone, but then at other site there was no difference. So we're still exploring the role that phenology plays, but clearly phenology can minimize pollen-based gene flow, so male gene flow in these systems, and has to be something that we have to consider. Okay, so conclusions. Tall and short form Spartina are ecotypes. That is, they're locally adapting their plant height and their above to below ground allocation. Our SNP survey suggests that tall and short form ecotypes have diverged repeatedly and in parallel. And this suggests to us that there's rapid evolution of local divergence in these ecologically important traits, and especially in the context of marsh resilience in the face of sea level rise, has implications for how we may manage these marshes moving forward. Okay. So I'm going to spend about 10 minutes then talking about our other story, sort of, if we could think of the Spartina story as a good consequence of rapid evolution, that is, it helps these marshes become more resilient. Rapid evolution in the context of an invasive species has bad consequences, because it, I would argue, means that these species are more able to tolerate the introduced ranges. All right, so there is a lot of homogenization going on, as you all are well aware. We are moving and have moved plants and animals all over the world, either deliberately or accidentally. This is as true for terrestrial species as it is for marine species. And One of the great questions for invasion biology, I would argue, is to what extent are the traits that underlie invasiveness canalized? Are they phenotypically plastic? Or is there actually rapid phenotypic evolution that also accompanies these invasions? And can we actually predict when and where it occurs? Well, when I first moved to South Carolina, I went out to these marshes and saw this, patches of a seaweed that's called a Garaphyton vermiculiphyllum. And as I started delving into it and looking at some of the older photos, I realized that this was an invasion that happened in South Carolina estuaries in about 2001, 2002. And it just exploded, not only here, but all over the world. So the first reports, we're still trying to get some herbarium specimens to try to figure out, but it's native to Japan. We've known that it's from Japan, Korea, Russia, and China, but it is now since the 80s in Europe, ranging from Morocco all the way up into the Baltic. We have recorded here on this coast from the Georgia-Florida border up into New Hampshire, and it's being recorded from Alaska all the way down to Baja, California. We don't see it yet in the Southern Hemisphere. Okay, well, a lot of people have been working on this system, including not just our lab, and there's a lot of traits about this thing that suggests it should be a great invader, right? So it turns out introduced populations have very strong antibacterial defenses, greater than those in the native range. They also have greater anti-herbivory defenses in the introduced range relative to the native range. They have a very flexible life cycle. And if anyone who thinks about mixed ploidy populations, this is a species that has a haploid and a diploid stage that are free living. And when I pick up a plant that's not reproductive, I can't tell the difference between whether it's a haploid or a diploid. And so that flexible life cycle actually has consequences for how well it succeeds in the invasive range. I'm not going to talk about that today, but it's a fun story. In some areas, it has a novel mutualism that helps facilitate its growth, especially in the Southeast. It has high genotypic richness, and so when it's in these patches, we know that richness enhances its growth rates. And then what I am going to talk about today is we believe that the populations in the introduced range have higher tolerance for abiotic stresses than the native populations. And thus, we believe that this phenotypic evolution helps facilitate the invasion. Okay, so we know from CO1 data, from microsatellite data, and from some unpublished SNP data that the source of this invasion is actually northeastern Japan. Now, northeastern Japan is pretty cold, so I want you to remember that. Northeastern Japan is also remarkable because it's a place where we actually think the vector is pretty clear, because northeastern Japan is the epicenter for oyster aquaculture. And after the Second World War, and even before the Second World War, Japan was a main producer of spat and subadults of Crassostrea gigas, which was transplanted all around the world. It is a fantastic market. There's something like $1.2 billion worth of oyster aquaculture happens today. And the primary vector for oysters that were sent to Europe and to Pacific Northwest, to California, to 6 of 7 continents around the world was this one region in Japan that's in the northeast of Japan, right? And so I went to visit there as a part of a project, and lo and behold, there's the Gracilaria vermiculiphylla. It looks exactly like the plant that we see in South Carolina. And genetically, it holds up. So we're still working with we actually have another grant focused on the invasion genetics of the Crassostrea gigas to be able to show that most of the invasion is from this area, as the historical anecdotes suggest. Okay, well, as I said, this is a cold area. And we were interested about whether when you transplant all of these plants from a cold area into the rest of the Northern Hemisphere, some of which are very warm, is there the possibility of a niche shift or a niche shift? So we did this analysis using sites that are confirmed by CO1 sequencing. We took some global databases that are out there, particularly those that are from BioOracle, and we made a niche model, right? So the niche model takes all of those variables for every one of these locations, and it says, well, okay, here's a axis PC1 that goes from cold areas to hot areas. And here's an axis PC2 that goes from lower variation areas, lower seasonal variation areas to higher seasonal variation. And if we just plot all of the native range locations and all of the non-native range locations, you know, actually what ends up happening is that it looks like all those native ranges have greater variation and they're spread between cold and hot areas. But when you actually focus just on the area where we know this invasion came from, right, the native source populations, then the picture changes. Those northern Japan samples are here in black, and they tend on average to be colder and more variable than most of the introduced range, which tends to be hotter, okay? Northern Japan feels a lot like and looks a lot like the Pacific Northwest, or even colder, especially when you get up to Hokkaido, the New England areas. Southern Japan has coral reefs, right? So the gradient in temperature is as profound along that Japanese coast as along this coast. So it's essentially, we're taking plants that were in these very cold areas and putting them in places like South Carolina and North Carolina, very warm. So that yields the possibility that there's been evolution in heat tolerance. Well, as part of a massive effort in 2015, our lab collected populations from Japan, West Coast, East Coast, and Europe. Clearly, this was something that we could do in person in 2015, could never do this today in person. We collected 15 native populations and 26 introduced populations, and we assayed them for heat tolerance, salt to cold tolerance, and some basically freshwater tolerance, how well they could survive freshwater. Now, this was all published in a 2018 Ecological Applications article, if you're interested. I guess what I will say is that our metric was bleaching score. So we collected these tips and we put them into the various treatments, and then we asked, let me forward this through here, and then every day we scored whether they bleached or not. And bleach means that they've lost their photosynthetic pigments, and so we know that that means that they lose their photosynthesis. And just to confirm that, when we had a fully intact plant with no bleaching, the photosynthetic efficiency that was measured by the PAM was very high, whereas when there was no bleaching, or sorry, fully bleached thalli had very low photosynthesis. Okay, I do want to acknowledge these fantastic scientists. They, over the course of about 4 months, recorded 74,000 bleach scores on over 1,000 individuals. It was an incredible team. They're all gone on to do great things, and I'm very, very grateful to them. Okay, so what did we find? We found that introduced populations have greater heat tolerance than do Japanese populations. Just to orient you to this plot, along the x-axis, we have the number of hours at 40 degrees Celsius, which is not a temperature that is unheard of in South Carolina. We get these temperatures on these mudflats, intertidal mudflats here. So during a low tide for 1 to 2 to 4 hours, you will see 40 degrees Celsius temperatures here. And the plants that we brought back from Japan were bleaching at a very high rate at the 4-hour mark. Whereas plants that we brought back from Europe or from the West Coast or brought from the East Coast, they bleached at a far lower rate. So clearly the Japanese populations are more susceptible to this heat stress. Now that was true for field collected. We also reared these plants in the common garden for about 3 to 4 months to try to minimize environmental effects. I know this is not a full generation that you'd like to see, but the fact that we still saw this pattern suggests that there is a genetic component to the greater heat tolerance among the non-native populations. Okay. That was not true just for heat stress. That was also true for cold stress. The Japanese population also had greater or less tolerance for cold stress. And the non-native populations were also more tolerant of low salinities. So all of this suggests that these populations in the non-native range are more hardy, consistently more hardy than the native source populations. All right. So why is that? Well, one reason might be that there's widespread rampant local selection, that in every place that these plants go to, they evolve and have evolved increased tolerance for these abiotic stresses. The other possibility is that during transplant, these plants, these genotypes were more likely to survive transport, and the transport in those oyster barrels may have been pretty stressful, elevated temperatures, maybe there's some fresh water and so on. And then finally, one that I think raises some questions for how we move forward with seaweed aquaculture, which is a growing industry worldwide, is that it may be that seaweed farmers in the past in Japan favored stress tolerant strains before introduction. And the reason I bring this up is that if you look at the old maps of where aquaculture of Gracilaria occurred, it was in these same places where these plants came from. So here's a figure from 1971. Every single one of these boxes represents a 20-ton mark per year of that seaweed. So an enormous amount in just above Tokyo and up in Hokkaido of Gracilaria aquaculture happened in these areas. We can't distinguish between historical selection and ongoing selection, but we're starting to think about ways to try to get into the historical herbaria specimens to try to tease this out. But I do think moving forward, this indicates that we need to be very careful about seaweed aquaculture because it could yield what we call super strains, analogously to what we've seen with some of the invasive species that evolved higher tolerance of abiotic and poor conditions inside of home aquaria, right? So Caulerpa is an example of a home aquaculture species that was released accidentally in the Mediterranean, and now it's a global invader. Okay, so conclusions. This seaweed invaded shorelines of the Northern Hemisphere, we think within the last 4 decades. It's since expanded rapidly within the last 10 to 20 years. We think the vector was aquacultured oysters, and we used some genetic tools to say that the source was in northeastern Japan. I'm going to skip over this, and I want to end with Charles Darwin, right? Just as I began, Charles is an inspiration every time I give this talk, every time I think about these questions. Charles should also be an inspiration we start thinking about managing for this good and bad evolution, right? So in the context of Spartina, what Charles would say is that we need to make sure that restoration efforts of these Spartina beds take into account local adaptation. And it's not only local adaptation of the marshes, but also within marsh variation in the environment. Charles would also tell us that if you have an invasive species you're worried about, try to keep it from coming in the first place, because rapid evolution is going to facilitate its success moving forward. And then finally, this point about seaweed aquaculture, Charles would say that we need to minimize evolution of these super strains, and there's a lot of really well understood mechanisms to do that, so that we don't run into more problems of seaweed aquaculture or some other manipulated species artificially selecting for species that might become invasive. So with that, I'd like to thank my collaborators on both projects. Again, Robin Zarabecki is the PhD who spearheaded a lot of the experiment. She's in Randall Hughes's lab and is co-advised with Tori Hanley. And then all of my collaborators involved with this Gracilaria effort. My former postdoc, Stacy Kruger-Hadfield, was a key component, as was Alan Strand. Rita Tirada, this wouldn't have happened without as well. So thank you for your time. And if we have any time, I'm happy to take questions.
Thank you very much, Dr. Sotka, for your excellent seminar. So we will open up to questions. Feel free, everyone, if you do have any questions, just unmute yourself and ask, or if you don't feel comfortable, you can, of course, write in the chat. So we will open up for questions. Yeah, take it away.
Can I ask one?
Of course, go ahead.
Hi, thanks for your talk. That was very interesting. I just wanted to ask at the beginning so that I don't forget what I was going to ask. So coming back to the Spartina, when you were showing the numbers of flowering shoots of the two types in the short area versus tall area zone, based on just number of flowering shoots, it looked like fitness of the tall should actually be higher in the short zone. But so what that got me thinking was, you did talk about flowering phenology in the context of gene flow, but I wonder if there is some genetic component to flowering time, and perhaps even more importantly to seed production, that might also explain, because it's all very well and good to flower, but if you're flowering at the wrong time for the short area, right, or producing seeds at the wrong time, maybe that would go some way to explaining the distribution across the zones.
So are you referring to this plot here where
yeah
the tall plants didn't flower as much in the short zone as did the short plants did, or the short ecotype flowered more frequently in the short zone?
Right, but what I mean is the tall ones flowered more in the short zone than in the tall zone.
The tall ones, let me get the highlight. So the tall genotype flowered less in the short zone than did the short genotype. And then the flip was also true, that the tall zone had a slight tendency to flower more in the tall zone than did the short genotype.
Right, but I was thinking from the perspective of just the tall genotype. The tall genotype actually flowers more, or has a higher proportion of flowering stems in the short zone.
Yes.
So it suggests to me that there's something else limiting it, which might potentially be around flowering timing or seed maturation timing or something like that.
Yeah, that's a great point. We didn't look in this experiment of when those two flowered in the short zone. And I think what you're saying is that there's a possibility that phenology may be operating not just between zones, but also within zones to minimize gene flow. That's a really interesting question. I thank you for that.
I have a question. So I think one just came in the chat, but I'll ask mine and then we can move over to the chat. So you said in one of your last points that there are a couple different known mechanisms in how to minimize the spread of some of these like super strains of these aquaculture. What are some of those known mechanisms, out of curiosity?
Yeah, so one of them is to keep these farms genetically diverse, right? So you don't just select for a single strain that is the most abundant, right? That we actually try to do a suite of strains. And part of that is because it's wise to have more diversity when you're trying to maintain a crop, right? You don't want disease to come in and wipe out your entire single genotype. The other is competition, and the other is recombination may prevent some of these basically growth, some of the growth to be able to get out of control. So there's genetic ways to be able to deal with it, to think about evolution specifically. I guess there's also sort of ecological and common sense things that you can do. For instance, you don't necessarily, you want to minimize the amount of feral release of these farms, right? So it depends on the crop. I mean, the kelps have to be placed, have to settle onto the benthos in order to be able to grow. So you know that they're going to stay in place unless there's some storm or some such event. But the gracilaria culturing is actually one that is by hand. It's a fragmenting industry. So In Chile, they actually farm Gracilaria by sticking the big clump of the Gracilaria into the sediment. And that what that does is, they don't need roots because they're seaweeds. But they grow and then they come back in another month and they'll take that clump, pick it up, split it apart, and put it into different clumps, right? It's a great way to be able to grow the Gracilaria, but it also means that you have a lot of fragmentation and you have these floating plants that could invade other plant areas as well. So maybe thinking of manipulations of the Gracilaria farming that minimizes the colonization of other habitats. That's one that that seems pretty straightforward to me. So there's what strains do we select for, and maybe we don't only plant that single best strain. We try to do a diversity of strains, and then there's also the notion of how we're doing these farms. We need to make sure that we minimize the release of them. That's a good question. Thank you.
Thanks. Okay, so one of our faculty members, Dr. Xenopoulos, says, great talk. And just a technical question was, unless you missed it, is bleaching just because of a loss in the chlorophyll pigments?
Chlorophylls, yes. There's also some red pigments whose name escapes me right now, but they're part of they're accessory pigments. So it's not just chlorophyll.
And so that's the purpose of bleaching, is to get rid of.
Well, the bleaching is, it's a stress response by the plant. It reflects the fact that they are either dead, either dead or just highly stressed. There are reports of plants being able to recolonize or regrow their pigments after they're released, but my sense from those studies is that the frequency by which they could just reproduce the pigments and just carry on is pretty low. So that, just like corals, when they bleach, there's a chance that they can get their zooxanthellae back and be able to survive. But it's certainly a low proportion of the population that can do that.
Do we have any other questions for Dr. Sotka? No? All right. Well, we can call that a wrap. So thanks again so much for tuning in today, everyone. And thanks again for delivering such an excellent seminar. Yeah, we really appreciate it.
All right, great. And I appreciate the invitation. I see there's a couple other chats, but thank you. Yes, yeah. Thank you for your time. I appreciate you listening.
Awesome. All right. Thanks, everyone.
Nice to meet you.